So, ladies and gentlemen, speaking of the very best, please welcome back Professor Brian Ford.
Today, we have an interesting talk in the sense that it's something you will never see on television. Nobody in the world will ever show you the things I'm going to show you today. You simply cannot find television programs that look at microbes. Now, the last program that I did where I actually looked at microbes on television was 30 years ago. And whenever I talk to producers, they always say, well, we have to fill in a form to show what the audience reaction is going to be before we can get the money for our program. And there aren't any programs about cells, so we can't give any indication of how much interest there is. The public isn't that interested in seeing living cells, so we can't really justify doing programs. And I say, but for heaven's sake, unless you show them the cells, how are they ever going to know what they're like? And they say, I know, it's terrible, isn't it? Anyway, about this other program. And they changed the subject. And I was sitting at home waiting to see a program where you actually saw some sort of intimation of a microbe. And eventually, a few weeks ago, I found one. And here it is.
Are you aware that you've exposed your skin to a whole host of airborne debeautifiers?
No.
This dermal magnifier will show the extent of the damage.
He's eating my beauty.
Try this rejuvenating lotion. It contains over 60 ingredients.
Whoa. I hope he didn't have children. Did he have children?
Millions of them.
And I also searched around and managed to find, in a Polish program, an advertisement that featured bacteria.
Dożywece rok. Używacie symbakterie.
Jasnie, szefie. Oni si myslí, že nás to zabíja.
My sa rozmnožujeme. Čo to tu vonia? Je to tak...
Osviežujúce.
Čože? Nový domestos Baktistop?
Nikdy som nič také osviežujúce necítil.
Vyblázni, veď nás to zabíja.
Nový domestos Baktistop likviduje baktérie a zároveň osviežuje.
Well, that's all you ever get about bacteria, I'm afraid. But this is what bacteria are actually like. These are little spiral bacteria filmed for me by my friend Bob Burden in Canada. And bacteria, of course, are very tiny, they're very simple, they have very little structure. But when you look at the larger cells, normal microbial cells, you do see some sites of incredible beauty. And I'm astonished that producers don't think people want to see these. Look at this glorious creature. It's a dinoflagellate called Serratium. And you see how it's going to swim with those two little whip-like flagellae. And do you notice that it's putting them out, it's not getting them tangled, it slowly unravels them, and it can then steer it any way that it wants to go. Here is Euplotes, one of my favourite organisms. Everybody says in microbiology, Euplotes, it whisks food into its gullet and it absorbs them and digests them. No, it doesn't. It selects what it wants, and it rejects those that it doesn't want to eat. They're very selective, and also under the microscope, incredibly beautiful. And this is the kind of creature that keeps water pure. When you see absolutely pure seawater or a transparent, crystal clear stream, you think, oh, there's no microbes in this. Yes, there are. There are these microbes growing on the rocks, and they comb all the bacteria out of the water and make it pure. And yet, time and time again, I get people saying, no, cells are terribly simple. Physics is on the point of solving everything. And I saw this the other day. Life is physics, it says. And it actually goes on to tell everybody that in next to no time, physicists will have a formula to explain everything about life. And if you look for a diagram for a living cell, then this is the kind of thing you find. It's just full of chemical formulae and chemical ideas, and it's just like some sort of organic transistor, and who's bothered about a thing like that? And if you look for living cells online, it's very hard to find them. If you do a Google search and you put in living cells and look for images, and these pictures come up, but virtually every one is a computer graphic. It's not a living cell. They keep creating views of living cells as though just little blobs of jelly. And many years ago, I was asked by the head of the BBC to propose a series of programs about cells. So I sent it in, And off we went to one of their producers, and later I discovered that, yes, the BBC was going to make a programme about cells, but they weren't going to involve me in it at all because they already had it all set up. So this was the way in which their programme started. And instead of beautiful living cells, all you saw were these blobs. I call them jelly beans. And everywhere you look in the media, all you see is jelly beans, which people label as living cells. Living cells are a lot more interesting than
The
this.
story of the cell is the most powerful story in science. We are cells.
That's computer graphics.
Cells do the work for us. It's the theory of everything.
It's absolutely astonishing. I mean, this is just a series of a couple of photographs taken under the scanning microscope, which they in the studio are moving apart. They're not really dividing at all. And you were stuck with this abiding image of the jelly bean. And people, I don't know why it is, people just won't show you or can't show you a living cell. Come to Stockholm, where the Wallenberg Foundation has funded research into living cells, and see what they make of them.
If scientists succeed in uncovering all the secrets of the cell, it can be a tremendous benefit for all humanity. Cell research is crucial to understanding how life works. The possibilities are enormous as scientists continue learning more and more about these building blocks of life. For the cell's life is part of our life.
But I have to tell you, that's not living cells. It's computer graphics. This isn't living cells either. Now, this is a sort of a simple documentary for the public, a fundraising documentary, really. So let's look at what senior academic students might learn in universities. And even here, when they're talking all about cells, they still can't show you cells, they only show you computer graphics.
Cellular processes can start or speed up or slow down, often leading to complex chains of events that make up the important processes in our biology. The cardiac gap junction and the natural killer cell examples both depend on the cells touching each other. But what if you wanted to talk to cells a little farther away? So paracrine signaling is a good way of communicating with nearby cells, but they can use some of the same mechanisms to talk to themselves. Next episode, we'll discover how our immune cells learned how to fight off invading pathogens and how they never forget.
Rabbit, rabbit, rabbit. All about living cells, but they never show you a living cell. These are not living cells, they're computer graphics. Somebody's just invented a round-looking blob and thought, let's stick it in. Here are people working with cells, and they cannot show you real living cells. Indeed, 50 years ago, I was caught by it myself when Harrop's, a publisher of mine in London, were publishing a book of mine about the microscope. And when the jacket came through, they had put one of these blobs on the front as well. So, I mean, I can't be too triumphalist about this because it's actually happened to me as well. Not long ago, I heard that a great friend of mine was going to put on a television program in which he was going to go out with a microscope in a boat and get some plankton and actually look at microbes. And everybody said, you've got to watch this program. So I copied it. I thought, this is great. I must have a video copy. I was so excited. But once again, living cells were virtually non-existent. Here's the program as it went out. And this is what we actually saw.
And then what we can do is we can just wash that out into a cup, and we can collect that to look at under the microscope later.
To actually see them, we're going to have to push our microscope camera to its very limits. What I really want to see, Emilian, is the phytoplankton.
Maybe we can zoom in on this one right here.
Oh, that's nice.
Yeah, they look like some diatoms. Oh, there he is.
Oh, that is beautiful. Fantastic.
And these last pictures, these last few frames, are actually 3 separate video films of dirt. One laid on, see how they're sliding against each other? They've got 3 films showing nothing but bits of dirt in the water, and by sliding them over each other, they made it look good. Jeremy Pickett-Heaps, who passed away recently, a friend of mine in Australia, he filmed plankton. This is what you should have seen This is what phytoplankton really look like. They are astonishingly beautiful. And yet that is something you're never going to see on TV. People say to me, can you see them in the electron microscope? And this is a picture of a cell under the transmission electron microscope with the nucleus and that dark nucleolus in the middle, and there's the cell wall. And you think to yourself, that's amazing. The detail you can see is extraordinary, but there are several problems with this. Firstly, it's only in black and white, and cells often have wonderful colors. Secondly, it's dead, and I am always interested in living cells. Thirdly, it's a thin, thin section, so you don't get any impression of what the cell itself is like in the round. Now, you can stain portions of cells in different colors and bring out the reality of a living cell. And if you want to have a look at that, then just look at this. This is taken by Matthew Daniels at the Wellcome Institute. That's a living cell stained to show all the tiny fibrils inside that cause it to move. And when cells divide, they look absolutely beautiful. And people often say to me, living cells are amazing, but you need a really good microscope to see them. But you don't. Living cells can just about be seen by the naked eye. And some cells are larger than others. And if you have a Tradescantia plant in your garden, then without a lens and with perfectly normal sight, if you look closely, you will see living cells. You can see, as we look closer, you see the little hairs around the stamens. Do you see they look like rather like a necklace? Each of those is a living cell, a single cell. And you can see them with the naked eye. And if you take one of those little hairs and put it under the microscope, you can see in the middle the nucleus. And around it, you can see the cytoplasm moving. Cytoplasm is the raw white of an egg. A hen's egg is a cell. It's got a calcareous shell, of course. But the white, the raw white, is the protoplasm of the cell, the cytoplasm. The yolk is its energy storage, and somewhere on the surface of the yolk is a tiny little nucleus which you can't see with the naked eye. But if you take a plant, any piece of plant, take a leaf and look at it under the microscope, and those little, with a lens, and those tiny little white dots you can see are the single cells. If you put it under a higher power microscope, of course, then you can see greater detail. And there is a single plant cell, a green plant cell, with the chloroplast, those green dots floating around. They are the little organelles that take solar energy and convert it into the production of carbohydrates from water and carbon dioxide. Humans can't do that. We cannot do this. That little weed growing in the crack in the sidewalk, that little tiny leaf, is using solar energy to create carbohydrates and proteins, basically speaking, out of air and water. We don't know how to do it. We think science is so good, but that little plant can do things we can't do. Some cells have a nucleus that goes wandering around the cell and patrols it as though on the lookout for problems. And of course, we all learned in school about the amoeba as the simplest form of life, a little shapeless blob of jelly, except that it knows where it's going, and it's composed of constituents that are soluble in water. But it doesn't just dissolve away. I mean, an amoeba is an extraordinary thing. The very fact that a simple blob of jelly can find its food and devour its food is an extraordinary thought. They're not simple at all. This is an amoeba. And every little particle inside that cell is performing a function. It knows what it's doing. It has its own sense of purpose. If we look under high power, and you can see wonderfully well how all of those tiny little particles are doing something. Because that little cell, in its own way, knows where it's going, knows what it's doing, knows what it wants to feed. And of course, we have amoebae inside our cells. We call them white cells. But the white cells in our blood are monitoring us all the time for disease. And if you inhale a bacterium, the chances are the white cell will find the bacteria and consume the bacteria and remove the bacteria before they can cause you any problems. Indeed, in 1959, this beautiful bit of film was taken by David Rogers at Vanderbilt University of a human white cell under time-lapse, and it's chasing those little black bacteria. And as you watch, you can see it sort of nosing its way around. This is a single living cell, but it knows what it's doing. It chases those bacteria till it collars them and eventually rounds on them and consumes them and dissolves them and destroys them. It's quite extraordinary to think of. More recently, Jeremy in Australia took this picture of leishmania, this little organism spread by sandflies, and it swims with the aid of this beautiful little flagellum at one end of its cell. There's a human white cell in the middle, and watch what it does to those two parasites. The one at the top is already just dissolving and being absorbed. And the one at the bottom, which would rather escape if it could, is attached to that white cell and broken down and dissolved and absorbed. Here, look at a couple more here. There's the leishmania cells, and look, the ones at the top, all being drawn into the white cell. Your white cells are doing this at the moment. At this very moment, you have inhaled some bacteria, and your white cells are eliminating them. There are 10 leishmania scattered around that white cell, and under this time-lapse film, all of them are inactivated by the white cell and absorbed by the white cell. It feeds on them. So that white cell actually metabolizes the parasites and controls them and eliminates them, and you stay healthy. And yet, you never find living cells on the telly. The BBC have a site where you can look for any form of life that you want, or so they say, and you can go scrolling down through all the various plants and animals, and you get simpler and simpler animals. You get right down towards the most lowly forms of life, and you end up with the earthworms, the Clitolata. And that's where you stop. Well, earthworms are quite big. Where are the microbes? They just don't exist in any program. And indeed, the BBC have become so obsessed with natural history that they never take it to the extreme and look at the microbes that actually make all other life possible. It's almost as though they've got a blind eye for it.
When I first started out, there was so much of the natural world we were unable to film. The cameras, the lighting, the size of the equipment, all meant that the extremely small, the very large, the ultra-fast, or the infinitesimally slow were beyond our reach. In the last 30 years, things have changed out of all recognition. We can now film everything, from the giants of the world's oceans to the smallest invertebrates.
Well, that small invertebrate, which my great and much admired friend David Atterbury is talking about, is a leafhopper, and it's made of about 5 million cells. But if only the BBC would just take a little drop of pond water and look at the extraordinary complexity of some of the single-celled microbes that surround us in our world, you would be utterly astonished. Isn't that an amazing sight? Just to watch a specialized single cell snuffling around in its environment. Here is one you may have actually come across yourself. This is the creature that makes sea glow in the dark. I don't know whether you've ever seen phosphorescent sea, but it's called Noctiluca. It has a single, sort of like a tentacle with which it catches other living organisms and absorbs them. And when it's disturbed in the dark sea, it sends out bursts of light, so the entire sea looks as though it's sparkling under the moonlight. It's an extraordinary sight. This particular cell has already captured a tiny little microbe, a little ciliated microbe, and is absorbing it as we film. And this is Coleps. It's sort of the scavenger of ponds. Each of these is a single-celled creature. But look at the complexity of it. Look at the structure of its cell wall. It looks almost as though it's tiled. It's beautifully made. And although it only contains cytoplasm, the same jelly-like material you get in a raw white of an egg, it specializes them so that they almost act like limbs. And it has no joints, it has no skeleton, it's got no muscles. And yet if you look at the bottom, you can see how those tiny little projections know exactly what they're doing. It's like watching a woodlouse. It's hard to imagine that this is just a single cell, but it manages to nuzzle around and to identify what it wants to eat and to open its mouth and to claw it in and to absorb it. And this is one living cell. In school, you may remember, I hope you remember, you came across paramecium. Here's paramecium. If you change the focus, you can actually get some impression of the delicate spiral sculpturing on the cell surface, which, do you see it there? It's rather wonderful. And there's its little beating oral groove with which it picks up the particles of living organisms around it that it wants to eat. And here's one of my favorites, stentor, shaped like a trumpet. And stentor is wonderful, but also it's selective. These little beating cilia, little hairs made of cytoplasm, set up a vortex in the water, and it picks out little organisms, but it rejects many of them. It doesn't think they're suitable for food. It decides what it wants to eat, and then when it finds what it wants to eat, it takes it in and absorbs it and digests it. The sheer beauty, this is blepharisma, the sheer beauty of some of these cells, this was photographed by Bob Burden in Canada. It's amazing to think that within a cell made purely of this gelatinous cytoplasm, you can possibly have anything so colourful, so complex, so beautifully choreographed in the way that it behaves, the way that it moves. It's astonishing to watch these things under the microscope. And here is Metopus. Again, it has so much complexity within one cell. And whenever I've shown these things to producers, they say, gosh, that's amazing. Never seen anything like that. And I say to them, no, you haven't, because it's never on the telly. On the other hand, it does mean that I can show it to people like you when I come and give a lecture. And it means that I alone have the chance to show these things, which nobody else is showing anybody. So from that point of view, it gives me lots of opportunities to show lots of beautiful pictures to lots of beautiful people. Here is somebody, a very eminent maker of computer games, making an elementary mistake that most ordinary people might make.
I'm going to show you a toy today that I've been working on that really I think of more than anything else as kind of a philosophy toy. In playing this toy, does it bring up philosophical questions in you? This game's called Spore. I've been working on it for several years. It's getting pretty close to finish now. And you actually start this game kind of in a drop of water. So here we are at a very kind of microscopic scale, swimming around. And I actually realized that cells don't have eyes, but it helps to make it cute.
Now, this is Will Wright, whose company is Electronic Arts. They make lots of leading computer games. And he says, I know that cells don't have eyes, but it makes it kind of cute. Oh, yes, they do. He puts eyes on his little microbe, but microbes do have eyes. Here is a microbe, and you can see that little red dot at the top. This is called Uroglena, and that little red dot is its eye spot. And if you take a section under the electron microscope and have a look, you can see it's got a curved retina, just like our eyes. But it can't be like our eyes because this is within a single cell. And here's a little swimming creature called Phacus, and it swims with this little flagellum. But you see, there's its little red eye spot. They do have eyes. Lots of microbes have eyes. Look at the front end of the cell. There you can see. Look at all the activity going on. And that little red spot is its eye spot. And sometimes when I'm working with these things, I get the strangest feeling as I'm watching them that they're looking straight back up at me. This is Eutropiella. And for some bizarre reason, it keeps pumping a blob of its cytoplasm from one end of its body to the other. Never quite known why it does that. Here we have a little organism called Euglena, snuffling around in a pond. And again, there's its little red eye spot. And Jeremy Pickett Heaps, bless him, took one of the most wonderful pictures I've ever seen of Euglena. Here is a single Euglena cell. Look at the structure of its cell wall. That's extraordinary, like a beautiful handbag. And you will see as it turns around, there's its eye spot, and there's its little mouth. And if you showed that to any child and said, what's that? The child, knowing nothing of living cells, would say, it's some sort of monster. There's its eye, there's its mouth. Although it's a single cell, we can still recognize what it is and what it's doing. Bob Burden took a lovely picture of all sorts of amoebae, and amoebae are remarkable. If you get a Petri dish and you've got lots of amoebae in it, and then you add lots of little ciliated organisms who are swimming around actively and leave it overnight, the most remarkable thing happens. When you come back the following day, the amoebae have eaten all the little ciliates. And nobody has any idea how they do it, because amoebae are just sitting around crawling slowly on the base, and the little ciliates are whizzing about like gnats on a warm evening. And Bob actually captured the end of an amoeba grabbing hold of one of those ciliates. He didn't catch the moment when it actually grabbed it, but he caught it just after, and that's a start. And if you look closely, you can see how the arms of that amoeba slowly move around to embrace and enclose and entrap that little ciliate, and eventually it will be absorbed and digested. Well, those are animal cells. What about plant cells? The round ones here are desmids, and the long ones are diatoms. And desmids, these beautiful desmids, this is called microsterias, and it's always described as being a sort of a round disc full of chlorophyll that collects solar energy, blah, blah, blah. But if you look at it closely, put it under the higher magnification, and you begin to get some sense of what's going on inside, and if you look at the tiniest details, just look at all the movement that's going on within. Do you see the little tiny particles moving in all directions, all with a sense of purpose, all carrying out some specific function? We don't know what any of these particles are. We don't know what they're composed of. We don't know what they do, but they do. And it's extraordinary to think, as you know, all cells reproduce by splitting in half, that something so complicated as that cell could ever divide. Watch at how it does it. And here's a picture taken by Jeremy, which shows, under time-lapse, one of these beautiful microsterious cells, and it's actually dividing in half as we watch. And every tiny little detailed part of that new half a cell forms with meticulous care until you end up with two beautifully formed cells where there was just one. It takes about 24 hours to do. And of course, this is greatly speeded up. But it's quite remarkable, isn't it, to think that such a complicated structure can ever reproduce itself. Well, there's the picture we saw before. We just looked at the round desmin. So what about the long diatoms? Well, diatoms have sex. Here are two pairs of diatoms. And if you watch very carefully, you will see that a little bulge appears on the side where they're nearly touching. And those little bulges become little tubes. And so the contents of the two diatoms can get together and actually form a cyst which will last throughout the winter. Isn't that an extraordinary thing? It's speeded up about 25 times. And you may also remember spirogyra from school. Well, here are two filaments of spirogyra, one at the top, one at the bottom. The one at the top is the male filament, the one at the bottom is the female filament. And under time-lapse, you can see how the male cells produce a tube that joins to the female filament. I mean, when people write about it, they say, a cellulose tube is formed that joins the two filaments together. Nobody has any idea how they do it. They have to actually recognize, first of all, that they're near each other. We don't know what the senses are. We don't know how they know that they're near each other. We've no idea how they manage to sculpt these beautiful little tubes that join the two of them together. But eventually, the male cells fuse with the female cells, and they round up and produce a solid cell wall and become a beautifully sound, hermetically sealed little spore that can outlast the winter. And some of them, of course, don't end up meeting their mate, the cells at the top, and they, I'm afraid, are doomed never to lose their virginity. Here's an organism called Oedogonium undergoing sexual reproduction. The little male cell has found a female ovum, and it attaches itself to the cell wall. It breaks a little hole, we don't know how, in the cell wall, and then it slowly migrates through so that it can contact the female cell, and it can then fuse with the female cell. And once again, this will produce a spore that will survive in winter when the pond dries up or it all freezes. So in springtime, a new plant will begin to form, a new group of colonies, a new group of cells. Do you remember Chlamydomonas? Everybody is aware of Chlamydomonas when you're in school. They have sex too. They will, in the autumn, meet each other, and they will swim around each other, and they will inspect each other, as though they can see whether they actually, as it were, fancy each other. And sometimes they'll just swim off, but occasionally they will remain pointing together like this, and they'll become closer, and eventually they will fuse, so you end up with a sort of a figure of eight-shaped organism with four flagella instead of just two with two, and that also will then form a spore that can survive during the winter. Cells reproduce by dividing in half, yes. But they also reproduce by having sex. And it's extraordinarily complicated to try and work out how these cells can detect what they're doing and how they actually perform what they do. Blepharisma. This is filmed by Bob Burden. Look at those two protozoan cells, those two ciliated cells. They've come together. You can almost see them in rapture as they conjugate. It's extraordinary to see that. Nobody would have any difficulty in realizing that these are two cells mating. It almost looks, and I'm trying not to be too anthropomorphic when I say this, it almost looks romantic. And yet, wherever you look, you see none of this wonderful choreographed complexity. All you see are those bloody jelly beans. Wherever you look, it's jelly beans. You never, ever see this incredible organization that real living cells embody.
One day, this lecture hall will be made of code. And though they're virtual students, what they'll learn together is real. The impact will be real.
What this student is holding is not real living cells at all. And yet, kids are seeing all this stuff and learning all this stuff. They're seeing all these computer graphic images. Nobody says to them, by the way, these aren't living cells. These are just simplified little drawings, like cartoons. And it remains a fact that the actual study of living cells is carried out by very few people and is, in many ways, the single area of science that nobody yet understands.
Aaron Bernstein, doctor and research associate at the Center for Health and the Global Environment at Harvard Medical School, has been studying the ties between the natural world and human health.
Really, we're utterly ignorant about the microbial world. In fact, it is the last great unexplored frontier in life on Earth.
And yet very few people are ever going to say that. I mean, looking down the microscope at these colpidium cells, I'm just astonished at how busy they are and how complex are their little lives. Here's that organism Ficus once again with its little red eye spot. I mean, some of these cells are just so exquisitely beautiful, purely as objects of aesthetic delight. And what about lacrimaria? There we are, there's lacrimaria, named like the lacrimal ducts, named because it looks like a teardrop. But look at it, nuzzling and nosing its way around. It's quite extraordinary, isn't it, to think that it can do that. Some of you may eat spirulina. Spirulina is a blue-green alga, and this is it under the microscope. And even something as simple as spirulina has its cytoplasm moving around inside the cells. Even something so terribly simple has life going on inside it. But when you look at something like Nasula, this incredibly complex-looking creature, it is amazing to think that nobody in the world can tell you what all those little dots are, what all those little particles are doing. You see, science has become obsessed with reductionism. Get a microscope and people automatically say, ooh, how much can it magnify? Give them an electron microscope and they'll say, that can magnify more than a light microscope can, so it must be much better. No, you don't study human sociology by putting a lens on somebody's forehead. You do it by standing back and looking how people interact. And I like looking at whole cells under low magnification. Sometimes you see the way that they behave, and they clearly have a sense of purpose. Here's a ciliate, it ignores that little diatom, but suddenly it senses another ciliate, a smaller one, and it noses around it before it gobbles it up and wolfs it in. It's like watching a cat catching a sparrow on the lawn, isn't it? It's quite extraordinary. The sheer ingenuity of some cells leaves my mind boggled.
Biologist and lecturer Dr. Brian J Ford having a long-lasting impact on science.
Well, he has done research on mad cow disease and even had laws changed because of his discoveries. Now, we had a chance to speak to him when he was in Singapore to deliver a distinguished visitors lecture at the Singapore Science Centre.
Well, one of the key studies that you have done, and that's made really headlines in the world of science, is intelligent cell. What did you mean by that?
Cells do extraordinary things. When we were in school, we all learned of amoebae as being the simplest, most lowly form of life. But there are some amoebae, and I'm doing studies of these at the moment in England. There are some amoebae that construct a home for themselves. They pick up things like tiny specks of sand and cement them into a beautiful vase-shaped house in which they live.
ever tells you that in school. You're told about the blob stuff. But there are many amoebae which actually make a shell for themselves. Here is an amoeboid shell. Here's another one shaped like a little purse, and you can see some tiny protozoan there. This is a dead and empty shell. You can see a little protozoan is nobbling around on the off chance of finding something. But here is one. It is vase-shaped, and it's called Nabila colaris, and that is the shell of an amoeba. The amoeba's not in it anymore, but these amoebae will actually pick up little sand grains and fit them together to make a shell. Now, you might think, caddisflies do that. I remember caddisfly larvae when I was a kid in a pond, yes. But a caddisfly makes a crappy, cruddy sort of a random-looking, messy shell for itself, and it's got a brain, it's got cement glands, it's got claws, it's got a skeleton, it's got nerves, it's got blood vessels. This is an amoeba, and it makes a beautiful little shell. And if you want an example of how clever you have to be to make a shell like that, then look at the kind of similar things that humans make. Because on a market stall, I found these little lampshades, and they're made in exactly the same way as the way in which an amoeba builds its shell. Here is one of those lampshades. Isn't it extraordinary? You can actually see there's a bit missing. I've never seen a bit missing when an amoeba makes its shell. But in actual fact, we can change this. You can see what an amoeba would have made for itself, because this is the amoeba's version. And it's exactly the same proportions and just as beautiful, and yet the amoeba does it with just those little pseudopodia. Simple. I don't think these shell-building amoebae are simple at all. In fact, I gave a lecture about them many years ago, and about 6 months later, I turned on the telly to find that they popped up on the television. And the result was quite extraordinary.
When an amoeba splits up, who gets the house?
Who gets the house?
Well, yes,
I thought the whole idea was that they split, they get two houses. There's some sort of perfect world of...
They assemble grains of sand, and they make that. That protects them.
Is that loads of grains of sand as they pull around?
Yeah, each one of those. It's about the size of a full stop on an average newspaper.
So, we've never heard that before.
That's a no,
that is actually quite interesting.
Hooray!
Why have we never heard of that?
I don't know. I suppose really.
They never mentioned that at school. There's been a conspiracy to keep this information from us.
Just listen again. I mean, Eddie Izzard was famous on this program for never being impressed by anything. And this is the first time that he'd ever said he thought that something was really, really interesting. And I love Bill Bailey's comment. No. He said, this is a conspiracy to keep this information from us. No, it's not a conspiracy. It's just ignorance on the part of producers. And they won't be brave enough to let me show what I'm being showing you.
That is actually quite interesting. We've never heard that before. Why have we never heard of that? They never mentioned that at school. There's been a conspiracy to keep this information from us.
Now, none of you, after seeing this, is going to doubt my contention that living cells are ingenious. The question is, are living cells intelligent? Now, there are many definitions of intelligence, but they all have the same point in them, and that is that an intelligent entity can find a solution to an unforeseeable problem. That's intelligence. Nothing to do with knowledge. You'll see people on quizzes coming out with amazing answers, and you say, ooh, how intelligent. No, that's not intelligence, that's knowledge. You can see somebody erecting a complicated building and say, ooh, how intelligent. No, they're following a plan, and they're doing something which is technically perfect. But that isn't intelligence either. Intelligence is when something that has never happened before happens, and when other people can't work out the answer, you can. That's when you're intelligent and they aren't. And I can show you an example from the lowly world of the red algae. Red algae, they're most commonly seen as seaweeds. Lava bread is made of red algae, the Welsh traditional food. When you go to the beach and you see those dark red, dark maroon clumps of plant life, those are red algae. They are so simple that no cell in the whole group of red algae can move by itself. They don't have any ciliated cells. They don't even have amoeboid cells. The cells just float around on the off chance of finding each other. They're very lowly. And my friend Jeremy had made a film, and he didn't realize its significance. What he did was this. He took a filament of these red algal cells and laid them on a slide, This, by the way, has been known to happen since Victorian times, but he's the only one I've known who filmed it. And you cut one of the cells in half, and all the cytoplasm spills out. It's just like cracking an egg and emptying it. And the question is, what happens next? Well, you'd think what happens next is that these two filaments would float away, and the cells would divide, and you'd get two new filaments for the price of one. No, no, no. Because it's on the microscopist's slide, these two broken cells remain next to each other. And nothing like this can ever have happened in history before. I mean, if a filament of an alga is snapped, it would be because a dinosaur trod on it, or there was a rock fall, or a sudden flood of water. And however it happened, the two broken ends would end up miles away from each other. The only place in the world where they can actually be broken and left touching each other is on this slide of a microscopist in his laboratory. And so here is Jeremy's cut filament. And you can see perfectly well, can't you, that the cell is broken and empty. But under time-lapse, look what happens. The two cells next door work out something has happened, and they've never done anything like this before. They change and expand in order to fill up the space that has been left by the cell being broken. Not only that, but as they're repairing the cell, they also repair the cell wall. Look, you can see how the cell wall is broken. It's like an L shape, like a Z, isn't it? But watch very closely. The broken cell wall, right in the middle of the picture, it slowly gets realigned, and then the cells create new cell wall material to seamlessly bridge the gap. So you end up with the cell that has been destroyed by this scientist completely rebuilt and functioning again because of the action of the two cells alongside. That's intelligence. That is intelligence in a simple alga. So never doubt that within cells there lurks intelligence. Indeed, on one occasion, I talked about it on the BBC, and their famous broadcaster, James Knockley, was reduced to silence, which has never happened to Knockley before or since.
Monday's Today programme, two basic laws of nature were rocked on their axes. First, eminent biologist Professor Brian Ford produced evidence that single cells can actually think. And second, and prepare yourself for this, James Knockley was actually lost for words at the thought. Here is that momentous encounter.
I'm very concerned that we have so retreated into things like molecular biology and genetics that we no longer stop to look at what entire cells do. If you culture brain cells in the lab and you pick up the electrical signals that they make a most irritating buzzing sound at about 40 hertz.
This is the first of a neuron spike, isn't it?
Yes.
Let's just hear it now.
Oh, ghastly.
Now, what was it doing?
Well, what the cells are doing is sending out little impulses, and we always believe, everybody knows this, that nerve cells are just transistors. They're a go or no-go gate, an on or off switch, a little binary code generating system, like a tiny transistor. Now, my view is that if amoebae are clever enough to build homes for themselves, it doesn't make sense that a neuron is only sending out no or no-go signals, on or off switch signals. So what I did was to take these standard spike recordings and to stretch them out from, I mean, you just heard about half a second, say 20 seconds, and at that level you can actually hear what is going on inside each spike. Each spike is more or less the frequency of the human voice. And so for the very first time, you can actually hear the utterances that one nerve cell is making to another. And this rather haunting sound is almost like, I think, seabirds on a cliff talking to each other.
Let's hear it. So what's going on?
Well, we always tend to teach these days that thought and data processing in the brain goes on at the synapses, the junctions between brain cells. I don't believe that. I believe that thought is going on within the nerve cells. The nerve cell is the most highly evolved cell that we know, and it cannot just be switching on or off like a transistor. I don't see us, you see, as bodies divided up into cells. I see us as great communities of independent, almost free-thinking cells that come together to produce the effect that we know of as human life. Well, Jim, you've gone suddenly almost speechless, which I have to say is uncharacteristic.
Well, no, I mean, after a description of what is the nature of life, it's a question of where you go next, really.
That stumped
Well, let
him,
me
didn't
help
it?
Jim out of his moment of scientific laryngitis.
That really did stump him. But that's the point. The sound that you heard there is the slowed down trace, played back as an audio signal, of the little electrical impulses that nerve cells make to each other. They're talking to each other. We're always told that thought goes on at the synapses, where it's just as though people see the neurons in your brain as being transistors and the brain as being like a computer. No, no, no. Each neuron, each separate cell inside your brain is the computer. They're thinking inside themselves, not between themselves. And when they have made up their minds about something, they send the signal to the next cell. Brains are 1 trillion times more complicated than we realize. The human brain is far more complex than anybody has begun to grasp. They still model it as though it was a computer. But no, each single cell inside your brain, if I'm right, is really the computer. So I hope you've enjoyed that little journey through these little simple amoebae that can actually make home for themselves, which, when a human craftsman makes them, we think is really quite clever. And yet, little amoebae can do that. And brain cells, if we listen to their signals in the right way, seem to be talking to each other, as though they're sending complicated messages. Believe me, ladies and gentlemen, living cells are miracles. I never cease from astonishment at looking at what they do. I'm so very honored that you could come today and share a little of it with me. And I hope that you'll take away some of those thoughts and possibly, if ever you get the opportunity, tell any producer friend you know that it's high time someone made a program or two about the wonder of the single living cell. Thank you.